This video is about enzymes. What are enzymes? Enzymes are biological catalysts that speed up metabolic reactions. Without enzymes, metabolic reactions would take place very slowly or not at all. So, what is a catalyst? A catalyst is a substance that increases the rate of a chemical reaction without being changed itself. So we can use the scissors model to understand the action of a catalyst or an enzyme. We use a pair of scissors to cut a paper into smaller pieces. So at the end, the paper changes, but the scissors remain unchanged. So the action of an enzyme is also similar to this. After speeding up the reaction, the molecules or the substrate can change into products, but the enzyme or the catalyst remains unchanged. All the enzymes are made up of protein molecules, and these enzymes are very specific to a particular substrate. Each enzyme has an active site that exactly fits its substrate. Substrate is a substance that the enzyme is acting on. The shape of the substrate is complementary to the shape of the active site so that they can perfectly fit. You can refer the diagram and see that the enzyme and substrate are complementary in shape. Actually, these are not 2D structures, they are 3D structures. As you can see in the diagram below, both enzyme and substrate have a 3D structure. Each enzyme can only act on a particular kind of substrate. For example, the enzyme amylase can act on starch molecules, but it cannot break down protein molecules because they do not fit into its active site. We can use the lock and key model to describe how each enzyme interacts with its specific substrate molecule. So we know that each lock has its specific key. So one lock cannot be unlocked by any other key. In the same way, the enzyme is like a lock into which a substrate molecule fits like a key. So as each lock has its specific key, each enzyme has its specific substrate. So this diagram helps us to understand how an enzyme works. So we can identify the enzyme molecule in blue color, and there are two substrate molecules, red and yellow. First, the substrate molecules slot into the active site of the enzyme. The enzyme substrate complex is formed, which is a temporary structure. The two substrate molecules then combine to form the product. Once the product is released, the enzyme remains unchanged and ready to accept substrate molecules again. So let's see what are the properties of enzymes. All enzymes are proteins. Enzymes are catalysts that are not changed in the chemical reactions. They can be used over and over again. Enzymes are specific. This means that each enzyme catalyzes only one kind of chemical reaction. Enzymes work best at a particular temperature, which is called the optimal temperature. So you can see the diagram. Enzymic activity is at its best at 40 degrees Celsius for this particular enzyme. Enzymes are made inactive by high temperature as protein molecules are denatured due to heat. Denaturing results in changing the shape of the active site. Enzymes work best at a particular pH. pH is a measure of how acidic or alkaline a solution is. Some enzymes work best in acidic conditions, which is low pH values, and others work best in neutral or alkaline conditions with high pH values. So according to the graph, the enzyme pepsin, which is a protease enzyme, works best at acidic pH levels, whereas most of the other enzymes, the optimum pH is around pH 7. Now let's see how enzyme activity changes with the temperature. According to the graph, we can see at very low temperatures, the rate of reaction is also very slow. So according to the particle theory, we know at low temperatures, molecules have less kinetic energy and they move slowly. Molecules collide less frequently and the rate of reaction is low. As the temperature rises, the rate of reaction increases. This is because at high temperatures, molecules have more kinetic energy and they collide more frequently. Then the chemical reactions happen faster at higher temperatures. The temperature at which the rate of reaction is the highest is called the optimum temperature. Different enzymes have different optimum temperatures. For example, enzymes from the human digestive system generally have an optimum temperature around 37 degrees Celsius. As the temperature increases beyond the optimum temperature, the rate of reaction decreases. At higher temperatures, the enzyme molecules start to lose their shape. The active site no longer fits perfectly with the substrate. The enzyme is said to be denatured. Denaturation is irreversible, which means once an enzyme is denatured, it cannot be reversed, which means denaturation is a permanent change. It can no longer catalyze the reaction. pH and enzyme activity. The pH of a solution affects the shape of an enzyme. Most enzymes are in their correct shape at a pH of about 7. In very high or low pH, enzymes get denatured. Thus, the active site no longer fits the substrate. So, the enzyme can no longer catalyze its reaction. Some enzymes have an optimum pH that is not neutral. Example, pepsin in the human stomach has an optimum pH of about 2 as hydrochloric acid is in our stomach. Now let's see some commercial uses of enzymes. First one is biological washing powders. Biological washing powders contain protease and lipase enzymes to remove protein stains and fat or grease from clothes very easily. For example, if there is a blood stain, blood contains hemoglobin, which is a protein. So when a biological washing powder is used, this hemoglobin is very easily broken down and the blood stain would be removed very easily. So biological washing powders are more effective than the detergent alone. But one thing we have to remember when we use biological washing powders, the water should not be very hot because we know that enzymes are denatured due to very high heat. So if very hot water is used, the enzymes will not be effective. The second use is extracting fruit juices using pectinase. Pectin is a substance which sticks plant cells together. Fruits like apple or orange contain a lot of pectin. So the enzyme pectinase is used to break down pectin because it increases the yield as well as it gives a clear and visually attractive final product. Enzymes like proteases are sometimes used when making baby foods. These enzymes enhance digestion and absorption of nutrients, making it easier for the infant. Another use of enzymes is in sugar syrup production, where amylase is used to break down starch to the sweet tasting disaccharide maltose.